Good afternoon, everybody. My name is Susan Hillier, and I'm the Dean for Research at University of South Australia in Allied Health and Human Performance. May I just take this opportunity firstly to acknowledge that I'm certainly meeting on the lands of the Kaurna people, and I pay my respects to elders past, present and emerging, and encourage you to acknowledge in your own way the various country and lands that you're meeting on as well. So it's always really great when this month comes around and I get to see some of my colleagues that I often don't see. So today is particularly terrific because I haven't seen Stuart for ages and first time meeting Tessa. So Professor Stuart Pitson will be known to many of you. Not that he's been around a long time, but let's say he's just started very young, Stuart. That's, I think, the way to say that, isn't it?
That's one way of saying it, yeah. May not be the truth, but
Stuart is a really, is an absolute key leader in the Centre for Cancer Biology. That's in the, as Tessa called it, the lovely fancy Bradley building in North Terrace up in the ABMC district. Stuart holds an NHMRC fellowship, and he also holds a chair in the Neurosurgical Research Foundation, or is it just Neurosurgical Foundation, no research? This is a really prestigious chair. This is actually relatively new, I think, if I remember rightly, in my 10 years past.
3 years.
3 years. Stuart particularly works in cell signalling, but being the chair in neurosurgery would tell you his particular interest is in brain tumour, and that's an area very close to my clinical heart. So good on you, Stuart. So I'll leave it at that, and I'll let you share your screen and look forward to hearing your presentation about current work.
Great, thanks, Susan. So I really wanted today to just talk about some of the brain tumour resources that we've developed in our lab. Obviously, we do a lot of brain tumour research, but we've also developed a number of brain tumour resources, which we think could be quite useful for the general scientific community in South Australia, and we're very open to collaborating in this space. So this is probably the best use of these 8 minutes, I think, is to highlight these resources that we have. So we do a range of different cancers we study in my lab, but we have a brain tumour research group which is headed by Dr. Melinda T and Dr. Bryony Glidden. And really in that group, we look at developing new approaches to treat glioblastoma, the most aggressive and common adult brain tumour, primary brain tumour. We also, so we look at targeting sphingolipid metabolism and looking at blood-brain barrier permeabilization as ways of targeting these tumours. What I want to talk about today is the development of advanced glioblastoma resources. We also study medulloblastoma, the most common childhood brain tumour, and we'll also hopefully get time at the end to show you a few little developments in medulloblastoma resources that we're bringing into South Australia. So really the overview of what I'm going to talk about is that we have managed to get access to a lot of patient tissue in South Australia. When I first started doing brain tumour research about 5 or 6 years ago, it was very limited in what we could access, and really people around here we're really using cell lines that have been around for 30 or so years, which don't really represent normal brain tumours anymore. So we managed to get access through the South Australian Neurological Tumour Bank, which is based down at Flinders, in particular Santosh Panus, a very dedicated neurosurgeon who's been helping facilitate this. So we're able to get patient tissue and Q's, I'll talk about that in a moment, which is the aspirate, the solidified aspirate of tumours from the surgeries. From that, we're able to make glioblastoma GNS cell lines, the glioma neural stem cell lines. From that, we're also then able to, sort of lost my pointer here, but we're able to then do intracranial tumour glioblastoma xenographs in mice. So I'll go through a little bit of those steps. We do a lot of genomic transcriptomic analysis of these cells and of the xenografts. We also do some heterotopic xenografts, so taking the tumour material straight into mice to try and maintain all of the proper properties of the tumours that may get lost in doing any in vitro culture. There's also quite a bit of work going on in the CCB in glioblastoma explants, so taking the tumour tissue, dicing it up under certain conditions and being able to grow that in the laboratory. That's mainly done with Guillermo Gomez and Michael Brown's labs. There's also, Guillermo has some glioblastoma organoid work as well. And with all of that, so this tissue gets segregated into various different labs. And really, we have these synergistic interactions between the various labs in the CCB to be able to use this tissue. And from that, so we are looking, at the moment, we've generated 52 low passage patient-derived glioblastoma cell lines. That encompasses the various different subtypes of glioblastoma. There's a number of different explants and organoids, 3 different heterotopic xenographs, and we've got 8 patient-derived xenographs that we use more routinely. So, and again, we're using those to look at efficacy of new therapies and understanding fundamental biology of glioblastoma. So very quickly, I'll whip through some of this. So essentially, we have the solid tumor. We get solid tumor, which is usually from the invasive edge of the tumor, and we also get the surgical aspirate from the ultrasonic probe, which is called the accuser, and that's normally from the core of the tumor. We use both of those to isolate tumor cells as in single cell suspensions and grow cultures out, these glioma neural stem cell cultures, which really are the stem cells that drive the tumor formation. We also get pathology reports and information about the patients, obviously not their identity, but information about their therapies and their survival. From to date, we've had 60 glioblastoma patient samples over the last 4 or 5 years. 48 of those have been primary tumors and 12 recurrent. From that, we've managed to generate 52 of these cell lines. Only one of those have actually managed to come from recurrent tumor. We're getting very good at getting cell lines from the primary tumors, but the recurrent ones are quite difficult to get cell lines out of. But we now have one of those at least, and we're hoping to generate more. Just looking at the breakdown of, there's fairly good cross section of ages and sex of the patients, and we can see here in this middle panel that we have tumours from different locations within the brain, which pretty much represents what normally happens, a cross-section of what happens in patients. So we're getting quite a good database of these samples, of these cell lines. As I said, we do quite a bit of molecular profiling and genomic analysis on these cells. We need a lot more, but it's quite expensive, of course. So we do a lot of RNA-seq analysis on the tumours, and this is just to remind me that essentially we can classify the tumours into the various 3 main subtypes of glioblastoma, which is the proneural, the mesenchymal, and the other one I can't seem to remember at the moment, and the classical proneural and mesenchymal are the 3. So we've got cell lines that essentially cover all of those different subtypes, and that's quite important because we can then start to look at personalised medicine for different subtypes. We've done quite a bit of drug screening with these cells as well. So this is just an example. We have a drug library, we developed, which is 145 different FDA, TGA approved drugs or drugs that are in late stage clinical trials. We screen them against various cell lines. In this case, it's a cell line, a U251 cell line, and then we've managed to cross-screen the most potent of those drugs against a couple of different patient-derived cells. We've now extended that out to another 20 or so cell lines, patient-derived cell lines. So we've got this screening capacity, at least with these 2D cultures of patient-derived cells, to be able to look at potency of different drugs. What you can tell from this, though, that most of the cells are not particularly sensitive to these drugs. That they're all screened at 100 nanomolar, but there's only really a few different drugs that will hit these types of cells. What I really want to talk about, though, is the patient-derived xenografts. So we've got a pretty good capacity now to be able to make these glioblastoma neural stem cell lines luciferase and GFP positive, so that we can implant them into the brains of mice. We use this a very advanced stereotactic setup to be able to very carefully implant them in exactly the same spot within each mouse. To give you an indication here, we put 50 to 200,000 cells into the exact location in the brain. That's in a 2 microliter volume over 4 minutes. So you really need these motorized stereotactic setups to be able to do this. There's no way you can do that by hand. And the reason you need to have such a small volume dispensed within over such a long period of time is that you need to make sure that you end up dissipating the liquid throughout the brain. Otherwise, what happens as soon as you remove the needle, the cells will come back through that needle shaft line that you put through the brain, and you'll end up with tumours growing in different parts of the brain, which is not appropriate. So here is just an image of mice that have a range of different patient-derived tumours growing in them, or a cell line is the first one. Here's the survival analysis of these mice harbouring these different tumours. So you can see we have, that's the red line here is a cell line, U87, which is the most common glioblastoma cell line used. But then we have a whole range of different patient-derived cells. So you can see some that are quite aggressive and advance quite strongly or quickly, others that take longer. But what is quite important here is that nearly all of them all the mice go down, within each set go down very similar time. So we have a very reproducible model, which means that we can actually do therapeutic interventions with a minimal amount of mice. So we get fairly good statistical analysis with a smaller number of mice. So they're very reliable and very consistent models. What is in, when we look at the tumours, you can see there's a whole range of different models here. So these are U87 and U251s in the middle here. You can see what these are cell lines. You can see they grow as distinct tumours, which have a very clear differential between the tumour and the surrounding normal brain. If you focus in here, you can see the H&E staining showing tumour in the darker purple and the normal brain in lighter pink. If you stain for human mitochondria, just as a way of staining for human cells, you can see a very distinct margin between human and mouse tissue for both these two cell lines. So they're not at all how a glioblastoma would grow in a patient. So we don't believe these cell lines are particularly good for modelling brain tumour in mice. Compared to that, we see here one patient-derived cell. You can see there's a much more diffuse margin where the tumour meets the normal brain, which is much more what would happen in a patient. And you can see here that most of the other tumours, over on the left-hand side, most of the other tumours, first of all, they take up a huge portion of the brain, but there's this very loose distinction between tumour and normal brain, which is exactly what happens with these glioblastoma patients. Some here, like this G23T, it will grow as a distinct tumour, but most of the others we've got are much more diffuse, again, which is just what happens in patients. We also have some syngeneic xenographs that, so mouse glioblastoma cell lines that we can implant into mice as well. Again, they're luciferase positive, and again, we see reasonably consistent clinical symptoms in these mice that again make them quite good models. And so really to wrap up, we've got a whole range of different models. We're putting those through a number of different approaches, so helping people to screen some drugs. We've got two different drug studies we've done. We've got a number of different immunotherapies studies we've done with Tessa and Michael Brown as well. And I guess the last thing I want to talk about is we also have started to bring in some medulloblastoma, this childhood brain tumour models coming through as well. So these are much more in their infancy, but we managed to get some models from the Fred Hutch Institute in Seattle. And again, we're just looking at a number of different medulloblastoma xenographs in mice. And again, we get some that actually advance very quickly and are quite good models to study therapies. So on that, I'll finish up because I think my time is up because Susan's reappeared in my screen. And I just want to thank various funding bodies, particularly the Neurosurgical Research Foundation for the support and HMRC as well. Very happy to take any questions.
Great. Thanks so much, Stuart. So if you have a question, you can pop it in the Q&A section, which I will just open up. While we're just waiting for the questions, Stuart, I know you're a really good collaborator. If people are interested in finding out more, what's the best way to
Absolutely, just contact me, email me, give me a call. We're developing these models to be able to collaborate with people. So we're very keen on collaborating with people. We can run, the best way of doing it is we run the models for people. And so people have got an idea of a new drug that they know is reasonably well tolerated in mice. Then we're happy to have a chat. And there's good evidence that might have a potency in these types of models. And we're very, very happy to work with people to actually run their drug through these models.
Fantastic. Great. Okay. So I really encourage people to get in touch. So let's now take, it's not a full turn, isn't it? Because we're sort of on the same theme. So I'm really also delighted to introduce you to Dr. Tessa Gargett. I don't know that I've said your last name right because I forgot to ask that. Tessa. We'll just call you Tessa. Dr. Tessa. who's an early career researcher. So it's great to have, can I say the old and the new, Stuart? Does that sound a little bit
it's the truth, so yes, okay.
The established and the up and coming. How about that? That's a bit nicer. So Tessa's an early career researcher at SA Health, but in the also in the CCB part in the Bradley building in North Terrace, if you need to find her. And her early career fellowship is funded by the Cancer Council. So that will give you a clue that her work is in cancer. So Tessa, if you want to start sharing your screen, and
sure.
I'll pass over to you. Thank you.
All right. So I'm going to dive straight in because I've got lots of data I want to show you and I don't want to run out of time. But I'm going to be talking about CAR T cells. And in my field in immunology, CAR T cells are so famous that I stopped giving a background to them because everyone was just getting annoyed. But I did slip in one diagram here just in case you're not familiar. So these are genetically engineered T cells. They have a new receptor that redirects their specificity towards a cancer cell so they can affect cancer cell killing. The reason they're so famous is because they've been very effective in treating CD19 positive B-cell lymphomas. So giving complete responses in those patients, leading to FDA approval, and they're just starting to come into the clinic here in Australia now. But what we're working with is a different CAR T cell with a different target. So we're looking at GD2, which I call a glycolipid, and Stuart always reminds me is a glycosphingolipid. But it's expressed on solid tumours. So we're not looking at hematological cancers like the B-cell lymphomas. We're looking at tumours like melanoma, and we've also been looking in some sarcoma patients for this trial. So the CARPETS trial is what I'm going to talk about first, and this is run by Professor Michael Brown at the Royal Adelaide Hospital, and I'm responsible for manufacturing the CAR T-cell product, SA Pathology. And it started off in melanoma patients, as I said, and then it sort of branched out into other cancer types. And so far, we've treated 12 solid cancer patients, and it's a very small trial, obviously, phase 1 trial. We're looking at safety primarily and feasibility. And fortunately, it's been very safe. So we haven't had any serious adverse events, none of the kind of cytokine storm that's associated with the CD19 CAR T cell treatment. But we haven't seen particularly spectacular responses either. So at best, when we give the CAR T cells alone, we see a stabilization in some of the patient's disease. So they had progressive disease, and then after the CAR T cell treatment, their disease stabilizes and stops growing over a number of months. So one of the reasons we think the CAR T cells aren't as effective as we'd hope is to do with the level of antigen expression. So I've put some tissue staining down there. You'll see the dark pink represents our target antigen, the GD2. And although some of the cells are very highly expressing GD2, there's plenty that are low or negative. So perhaps these cancer patients are not the best group for us to be trialling the therapy in. And so we've turned our attention to primary brain cancer. So particularly glioblastoma and a diffuse midline glioma that is in children. And we've been using this tumour bank that Stuart's introduced to you to screen patients' primary tissues for GD2. We found very high levels of GD2 expression. And it had already been reported in the childhood cancer that there was high levels of GD2. So we thought this represented a better target population for our therapy. And we also see high GD2 expression in those cell lines that we grow out of the tumour material, which Stuart also described. So what we're now going to move on to is some data from preclinical testing in mouse models. So we're using the intracranial model that Stuart's Pitson's group runs for us. So they do the surgeries and then we do the CAR T cell treatment. And one of the nice things from looking at these mice is the tumour retains that really high GD2 expression, and it's very much associated with the tumour and not the healthy mouse brain. So a kind of key point I need to make here is the GD2 epitope is conserved. So it's identical in human and mice, which means we can make an initial assessment of toxicity. One of the concerns about this CAR T cell treatment is that it will target the healthy brain tissue that has very low levels of GD2 expression, leading to neurotoxicity. But we haven't seen any evidence of that in the mice. The mice who are treated with CAR T cells actually do much better than the tumour-bearing untreated mice. And we don't see any neurological signs that they're experiencing neuroinflammation. And we've also sent the tissues to independent pathologists, and they've reported that in the healthy side of the brain not occupied by the tumour, there's no evidence of CAR T cell infiltration there or of damage to the healthy tissue. So we're also using this model to try and investigate the factors that lead to CAR T-cell control of tumour versus tumour escape. And before I move on, I will point out that we've treated mice with CAR T-cells derived both from healthy volunteer blood and from GBM patient blood, because we were concerned that the GBM patients' T-cells might not be suitable to make a CAR T-cell product, which was a important point that needed to be tested before we could look at clinical trials. But fortunately, the GBM-derived CAR T-cells, which were represented in the blue there, were equally effective at controlling the tumour. And obviously, you can see from the survival curves, it's a short-term control, so we do get a survival benefit, but the tumour does regrow, and eventually the mice have to be euthanized. So one of the other analyses we're doing in this experiment is looking at whole brain sections. So we can look at sort of the spatial components of what's happening in the tumour and what happens when we treat with the CAR T cells. So again, here I've got the tumour highlighted with GD2 staining in pink. And then I'm going to show a brain from a mouse that was treated with CAR T cells. So the CAR T cells are represented in green. And so I found this really striking, this sort of mass infiltration of T cells. You'll notice that the red staining, the GD2 staining, is significantly reduced. There are still some GD2 positive cells there, so some residual tumour, but most of the tumour has been destroyed by the CAR T cells. And importantly, these mice didn't show any neurological signs despite this mass infiltration of CAR T cells in their brain. They were actually euthanized at endpoint due to poor body condition and weight loss, which we think is evidence of a kind of graft versus host disease, because these are immune suppressed mice that are receiving human T cells. So it's more of a reaction to the treatment than the tumour. So I want to just mention this very briefly. The CAR T-cell technology we're using in the clinic is sort of 10 years old now, and people are continually refining and optimizing CAR technology in the lab. So we've able to access one of these kind of newer technologies where the CAR T-cells also make their own IL-15. And we found that those CAR T-cells are basically even better. So we got 50% of mice at the end of the experiment were still alive with no neurological symptoms and had very, very low tumour burden, undetectable in some mice. And you'll probably notice there the CAR T-cell infiltration pattern was quite different. We didn't get such large numbers of the CAR T-cells in the brain, and yet they were equally effective at controlling the tumour. So we think this is a promising candidate for future round of clinical trials. I also highlighted the issue of tumour escape, so the tumour rebounding after treatment. And we've investigated this a little bit. So this is showing a comparison between GD2 staining, which is our target antigen, and human GFAP staining. So in an untreated mouse at the top there's good correlation between GD2 and GFAP. But then in a CAR T-cell treated mouse, if you just look at the GD2 staining, it looks very promising, like there's no GD2 positive cancer left. But in fact, that whole space is occupied by a GD2 negative GFAP positive tumour. So that's a classic example of the tumour downregulating the target antigen and then continuing to grow, so escaping the control of the CAR T cells. But promisingly, the IL-15 CAR T cells, so this newer generation technology, we don't see tumour escape to the same extent. There is some GFAP staining there, and they do have a small residual tumour that will probably start to regrow, but it seems to be happening much more slowly than for the older generation technology. And I'm just going to finish up by talking about the clinical trials that are sort of imminent. So this is, again, using the older generation technology, but we have 2 approved from the TGA. There'll be a paediatric trial run by David Ziegler at the Sydney Children's Hospital, and an adult GBM trial run by Michael Brown at the Royal Adelaide Hospital, and hopefully Flinders as well. But all the manufacturing will be done at a single site by my team at SA Pathology. So we'll be shipping the CAR T cells to Sydney. So we're really excited to get HREC approval in March this year. So that's for the paediatric trial. So that'll be opening really soon. Unfortunately, the adult GBM trial is lagging behind a bit, mostly because they're trying to work out in the protocol where the window of opportunity is to treat these patients, because their disease progresses so rapidly. They sort of have steroids, then surgery, then radiotherapy, then chemo, and they stay on chemo until they have a recurrence. So we're just trying to work out when is the best point to administer the CAR T cells. So that's underway at the moment. So I'll stop there. I just want to acknowledge all the wonderful lab staff who contribute to this project, and obviously Stuart's group who do all the tricky surgery for us, and the clinical staff who run the clinical side of things. We've got some really good clinical trials managers who help out with all the logistics. And a big shout out to the patients and their carers who donate this material, because it's so critical to our research. And I often think that they must be sick of being stuck with needles and not want to donate anymore. And yet they're very generous and they give us the blood and they agree to donate their tumour material. So we couldn't do it without them. That's it.
Great. That's a really important acknowledgement, Tessa. Thank you for that. So again, we'll just check on if you've got a question, pop it in the Q&A section. And while we're waiting for that, I might just pose you a question, Tessa, about the sort of toxicity aspects of what you're doing, and how are you investigating those sorts of, considering those sorts of issues?
Yeah, it's really tricky. So we can get an initial indication from the mice because they do share the antigen. But until we go into a human with a fully functional immune system, because these are immune deficient mice, we don't really know. And so we're going to have some very careful monitoring in place for the patients. So the kids in particular are going to stay in hospital for 7 days, because even if the treatment's working and you're not getting off target effects, where you're targeting healthy tissue, the destruction of the tumour cells in the brain will lead to inflammation, and we have to be able to manage that inflammation. When we know from the lymphoma trials that a lot of those patients did end up in ICU and had to be, very aggressively managed to bring the kind of systemic immune response that's happening under control. So that's something that the clinicians have to worry about and not me, but I know they've got a lot of protocols in place for what they're going to do in the event of, neurological symptoms in the patients.
Cool. Okay. So as usual, we've got a pretty shy audience. That's fine. So actually, Tess, would you just mind giving us a bit of what's your background? Oh, hang on, we've got one. Hi, Tessa. It's from Mark Bunting. Are you seeing CAR T activation and then exhaustion, which accounts for the less than ideal responses with the first generation cells?
Yes, most likely. I think there's a lot of things going on. I think they're struggling to get into these big solid tumours. So in the patients, they're at advanced stage and they have lots of metastatic lesions through it. So that, they have lung mets and liver mets. I just think it's a really big ask for the CAR T cells to get to all those sites and control the tumour. But equally, I think that all the normal immune suppressive mechanisms within the tumour are shutting down the CAR T cells. They're sort of superpowered. They can overcome some normal suppressive signals and provide their own activation, but they're not completely immune to things like PD-L1 signaling, and so I think they are getting turned off in the tumour, and it will rely on the next generation of technology, I think, to deal with those issues and some better genetic engineering to overcome those problems. Great.
Okay. Well, my question was going to be: are you really an early career researcher? Because that was a great presentation. And
I'm actually stretching the truth. I might be counted as big career now, but I still have my early career fellowship, which is
if you've still got your early career fellowship, just take it, yeah. Because I think that was we expect Stuart to do a fantastic job because he's been doing it for a while, but fantastic job and really clear presentation. So thank you. Like, I'm saying that from the position of someone who's not in this area at all. So I really thank both of you for your time today. I really encourage people who are listening now, who are listening in the future, to get in touch with either Tessa or Stuart. If you've got more questions or you want to collaborate, I can certainly confirm they are great to collaborate with. So please do keep in touch, and wish you a very pleasant evening. Thanks, everybody.
Thanks, Susan.
Thanks, Susan. Thanks, everybody.
Bye.
Bye.